So I would just like to acknowledge that we are uninvited guests on the unceded ancestral territories of the Musqueam, Squamish, and Tsleil-Waututh nations. And as the WHRI's Provincial Research Institute committed to improving the health outcomes of women, including those across the 2S plus spectrum, we would like to recognize our responsibility in the collective efforts towards establishing culturally safe healthcare systems and services that address health inequities among Indigenous peoples, especially Indigenous women, girls, and Two-Spirit peoples. And as we gather together today, we encourage you to reflect on your own positionality on these lands and your personal commitments to reconciliation. And I think this also offers an opportunity for us to reflect on the ways that we as a research community aim to meaningfully include the voices of Indigenous peoples and traditional ways of knowing in our work. So with that, I'm very pleased to be able to introduce our round speaker for today. So Dr. David Huntsman is a pathologist and a clinical molecular geneticist at UBC and is the Canada Research Chair in Molecular and Genomic Pathology. Dr. Huntsman directs OVCare at part of BC's multidisciplinary gynecologic cancer initiative and conducts his research at Vancouver General Hospital and the BC Cancer Agency. Dr. Huntsman's research has led to the development of diagnostic, predictive, and prognostic tissue-based cancer biomarkers for ovarian cancer and a wide variety of other tumor types. His team created a blueprint for subtype-specific ovarian cancer control and have been leaders in the application of novel genomic technologies to better prevent, diagnose, and treat ovarian cancer. Dr. Huntsman is also a fellow of the Royal Society of Canada and the Canadian Academy of Health Sciences. So with that, I am excited to turn the floor over to Dr. Huntsman.
Well, so can you hear me?
Yes.
Okay, great. So, well, first of all, it's a great honor to be here and to present, and I really hope some new collaborations will um, come from this and some new perspectives on the work I'll be presenting. So I'm going to be giving two presentations, and this may be a little bit like that Everything All at Once film from a couple of years ago, but I hope you don't feel shell-shocked by what's thrown at you. But there are two themes which I'm going to address, and well, let's see. Before that, I'd like to, of course, acknowledge that I'm sitting in my house, which near the university, which is on the traditional ancestral and unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. The outline I presented already in the poster, but just as important, or more importantly, some declarations. First of all, I am in a conflict of interest. I do offer advice to a genomic testing company. But more importantly, all work presented reflects the efforts of teams over the years, which includes colleagues, collaborators, trainees, technologists, and of course, our community and patient partners who really have enriched our efforts. I'd like to thank my team and funders, many fantastic grad students over the years who sort of built this work, and the collaborators in MAPcore, and also in OVCare, which has been an incredibly stimulating place to spend the last 20 years or so. So, in robust health research environments, and in particular, I think this is an opportunity to build on in BC, um, particularly as they're embedded within healthcare systems. There are programs, and programs are thematic in that they're coalesced around major health challenges and they focus on many aspects of a problem, and they use research cores to expedite their research. And OVCare, the GCI, would be an example of such a program. There are many others in the cancer space and other places. There are also cores which provide very specific, leading edge, scalable capacity and expertise to address a single aspect of many problems. And they serve many research programs. And if they're not serving many research programs, they're not a core and they probably shouldn't exist. And MAPcore would be an example of that. And I'm going to try to give the stories of both of those. So around the time I finished my research fellowship in the UK, the gene expression profiling world opened up with the papers from Chuck Peru and Pat Brown from Stanford. And it basically showed that if you grind up tissue samples and you look at the gene expression profiling, you can see order beyond what you could see on an H&E slide or by looking for an occasional mutation. And this did allow a clinically relevant classification of breast cancer, but surprisingly little else, even though there were lots of publications came from such work. But with the plethora of publications, there became the need to validate findings. And we were, myself, Blake Jolks, and Torsten Nilsson, were stimulated by a editorial in the New England Journal of Medicine talking about how there was a global roadblock in biomarker validation. And we realized we had a massive tissue archive in the VGH and large, well annotated outcome data in the Cancer Agency. And if we combined the both of them, we could really help in the effort to figure out what biomarkers could be used clinically and which couldn't. Um, and several of the people on this call, like Jeff Terry, did his PhD working in that space. Um, and that was GPEC, Genetic Pathology Evaluation Center. The picture's kind of amusing because when I found this, I realized, oh my gosh, that was a long time ago, and I didn't realize I was so short. The GPEC enabling technology was something called tissue microarrays, where instead of if you wanted to look at 1,000 cases for 10 biomarkers, you would take 10 sections from each block and do 10,000 stainings. You could put them into these recipient tissue microarrays and look at a large number of cases in a single slide. And we got very good at doing this work for immunohistochemistry, for fish, and for other methodologies. And that work went on quite happily, and we did some good stuff. And then something truly disruptive, for me, was profoundly exciting. It was next generation sequencing, and because of the work Marco Mara and Steve Jones had done on looking at SAGE and other very small fragment mapping projects, they were very well equipped to deal with the first next-gen sequencing data, which was very short read and poor quality, but it was beyond anything we could have dreamt of before. And so we kind of went all in on this. Um, we made a bunch of discoveries, and at the time I was quite filled with hubris and probably quite intolerable when I was giving lectures because I really thought finding the key mutations in cancers would lead to rapid progress. It actually did lead to almost instantaneous development of diagnostics, but very little happened afterwards. For instance, with FOXL2, we have no FOXL2 directed therapies in granulosa cell tumors because we don't have any model systems to study it. ARID1A is still a bit of a mess. In DICER, we've made some progress, which I'll talk about. And SMARCA4 actually made more progress, even though it's a much rarer cancer, small cell hypercalcemic ovarian cancers, but because we have many model systems. And the challenge there is there are so many ideas for new treatments, there aren't enough patients to try them in. So the initial manifestations of NGS have had a massive impact on diagnostics. So in the here and now, we do panel tests for germline and somatic mutation, exomes and genomes for broader views, ctDNA analysis for diagnosis and monitoring, but there's some missing elements. Cell context, spatial relationships, intratumoral heterogeneity, and the challenge to study very, very small lesions, where when you've got them, by the time you've seen them on a microscope slide, there's nothing left of the tissue. It's done. And also it's performed apart from the diagnostic flow, and there's very little integration. Proteomics has the similar challenges to date. Um, so how to bridge this? Liquid phase with in situ analysis. Well, LCM can be done, and we're actually investing more in this to support very low input proteomics. And this work's been done with Greg Moran and Philip Lang, Leonard Foster, and others. Single cell genomics um, tells you what's going on in a sample, but you don't have any spatial orientation. And we needed other technologies. So some on-slide analysis I really embraced, and I thought it would uh, something which pathology and other interested trainees might want to get into because it did tie to some of our core strengths. I think spatial context is essential for a meaningful analysis of tumor microenvironment. It's required to study lesions which are destroyed through their identification. And what's really exciting at this moment, there are some incredible new technologies which are converging, which we've managed to access. And I'll discuss a few, but it doesn't mean the ones which I'm not discussing do not have value. So how to study samples in depth on a slide without losing spatial context? Well, you can, in transcriptomics, you can do an unbiased analysis, and there are several approaches now where you can derive a sequencing library which is spatially orientated. And Visium was the first methodology, but Illumina now have a system, and BGI have a system which should be cheaper. I don't know if they're better. I have not seen data, but it gives a very detailed view with sequencing as a readout and barcodes, which are spatially orientated as the means to reconstruct the spatial relationships. The second methodology is probe-based, which tend to be where the capture methods tend to be optical, and you actually get the spatial orientation as part of the process. And there are many different methods here. Cosmics, but from nanostring, is used by many. We're using Xenium, and I will show data from that. And there are others emerging as well, and they all seem to have their upsides. So in the world of Xenium, it's one of two technologies which comes from 10x Genomics. Visium was the first uh, spatial system which was based on spatially aligned sequencing libraries. And Xenium is a commercialization of a process called MERFISH, where they have gene-specific padlock probes, which are put onto the slide, followed by cycles of rolling amplification and detection by fluorescent probes. Um, the methods are shown here. It takes a long time, it's expensive, but at the end of the day, whether you're looking at 300 gene panels in depth to look at what's going on in your slide, or the beautiful picture of some fallopian tube shown is from a 5,000 gene assay. It really, you can start seeing biology you didn't know existed before. Other methods are needed to validate findings, but it opens doors, and particularly doors which you can only open if you understand spatial relationships. And I'm thinking that some, in studies of placenta and certainly endometriosis, which I'm doing right now, with Paul and others, this is a really key technology. So we've been using this for many types of research. We've become interested in why some fallopian tubes or some women's, while some individuals are more prone to getting high-grade serous cancer, which comes from the fallopian tubes than others. And because when we look down a microscope at fallopian tube epithelium, we can just see secretory and ciliated cells. We can't really see what's going on. So this is an ideal opportunity to dig deeply into different cell populations, which are cryptic cell populations, and their roles in the development of this fallopian tube derived cancer. We can also start studying rare cancers. Mesonephric cancers come from the cervix, and they are connected to mesonephric remnants. But there's a mesonephric-like carcinoma which shares many features, which occurs in the uterus, where there are not mesonephric remnants. We've recently done a study where we could look at this. Now, you could use other methods to study the cancers themselves, but when it comes time to studying the epithelium of the mesonephric remnants, they're just too small to really study by any other method. But here we can now start getting these in-depth views. Then you can take all of the cells out of there and then start looking at what's going on in the different cell populations in exactly the same way you would study single-cell RNA-seq. But the key thing is every cell which you're studying can then get mapped spatially back to your slides. We're also using this to study humanized mouse xenografts to see what the human immune cell infiltrate is. And these xenografts are going to be key for us studying immune therapy combinations. What really surprised us in our version of the humanized mouse xenografts, there's many different flavors, these blue cells here are actually natural killer cells. And we were quite shocked to find that a large number of our infiltrating humans' inflammatory cells in the tumors in mice are actually NK cells, which it means this might be a better model for studying NK targeted therapies than perhaps T cell engagers. For proteins, again, two flavors. One is unbiased analysis, like the unbiased sequence analysis. And this involves taking a slide and slowly ablating what's on it and capturing everything from mass spec. This is slow, difficult, but unbiased. As mass specs get better, it may become more and more feasible. There's only a couple of labs in the world which do this. Um, and then there are antibody-based methods. As everyone on the call would know, there are some proteins you can't make good antibodies for. And also for secreted proteins, using antibodies is always a mess. So the unbiased analysis would be really useful, or combining looking at proteins with antibodies with some sort of RNA in situ. There are many different flavors of this. Opal was the old method from 15 years ago, got us up to about 8 antibodies. Orion is about 20 antibodies of one-shot analysis. You can do 3D microscopy with about 15 antibodies on some confocals. And then we move into some of the larger scale systems. So imaging mass spec can get you, almost to 100 antibodies. And then there's the Codex, PhenoCycler, and cyclic IF. And I'll talk a bit more about cyclic IF because it's the one we have in hand. But starting with imaging mass, this is Opal, which gets you to about 8 slides. It's really good, but it's not that efficient, and I don't think many people will be using it going into the future. Imaging mass spec was really exciting, where you increase the numbers of signals which you could tie to your antibodies by attaching metal ions to them. You put those on your slide, and then you use mass spec to identify what metals are in which place in the slide, and then build your spatial map from that. This does work. However, it's expensive. You can only look at small areas of tissue, and labeling novel antibodies or the antibody you're interested in with metal ions can sometimes prove to be impossible. So it's not practical. And a lot of people with these systems are now kind of sliding away from them because it's just too hard. The PhenoCycler came out from MAPcore, and it is an effective kind of cyclic analysis of many epitopes used by using antibodies which are labeled with DNA barcodes. This works, and it can work really well for established labeled antibodies. And it's just some pictures from the core in Victoria. The challenge is labeling new antibodies with DNA tags is difficult, and it's expensive as well. We went for cyclic IF, which is conceptually quite simple. So rather than increasing the number of signals you use the same small number again and again and again. And between each round, you erase the signals. And it tends to use, all the systems use standard labeled or even sometimes unlabeled antibodies. It's completely flexible. You can combine any antibodies, and there are academic and commercial systems. And these are just some images from our system. And the one on the right, I think, is just particularly beautiful, the ovarian cancer and the immune infiltrates and stroma around it. So this is kind of cyclic IF again. The Between each round of staining, there's QA checks, there's correction for autofluorescence, and we tend to be able to build up very high quality images. Because it's cyclical, there are many repeated steps, and it takes a bit of time. We can do 8 antibodies a day on a section, and I think we can do 8 slides at a time. So we slowly build up these sort of composite images. One system which exists is the Comet Lunaphore system. It has a microfluidics chip. It uses unlabeled antibodies, which is kind of interesting. The challenge is it only analyzes a small region on a slide. And we have a massive legacy tissue array archive, and it wasn't going to work for us, so we moved away from that. Maxima is an interesting system from a German company, but it only works on half of a glass slide, a standard pathology slide. And again, it was going to be proved too expensive for us, even though the images they produce look great. So we opted for the cell dive from Leica. It's built off an old GE platform, which they purchased. We bought a liquid handling robot, which goes alongside. We like it because there's no disposable microfluidics. Antibodies are basically the big cost. We've gone up to around 60 antibodies. We don't know what the maximum is, but I don't think that's ever going to really be needed. Whole slide. very high resolution. So we can actually look at DNA damage foci in nuclei and things like this, again, spatially connected to spatial information. And you can run some antibodies, put it in a fridge, take it out, add a few more to the same kind of composite image. Um, this is the workflow. Um, and it again, this runs 24/7. We have a fantastic team who've operationalized this. And in the cancer context, it provides spatial visualization of complex pathologies. We're using this as our show to look at ADC targets and to validate transcriptomic targets on a protein level. Just a sense of what you can see. This is an endometrial cancer. This is the sort of image we've seen in the last 150 years, where you can't really see what's going on in the stroma, and also you can't appreciate how heterogeneous the tumor is. On the cell dive, we can see about half of the cancer cells have pink membranes, meaning they're HER2 positive, and the parts of the cancer which aren't HER2 positive, a lot of them have blue nuclei showing that they're ER positive. And then there's some parts of the cancer which are positive for Neva marker, which is kind of really interesting. And then there's a very robust and diverse immune infiltrate. We're working across cancers and across diseases to build up a large panel of ADC targets, which we can start applying to rare and common cancers to determine which combinations of ADCs or antibody drug conjugates which could be used, and for rare cancers, which would be the best ADCs to take into clinical trial. For gynae research, detailed analysis of cancers, precursors, at-risk tissues connected to relevant questions, detailed analysis of tumor microenvironment and other microenvironments, discovery of cryptic cell populations, such as we're starting to see diversity in secretory cells in the fallopian tube, which we weren't able to appreciate before, and these may have real relevance, particularly for the work we're doing with Dr. Norman, who's I saw is on the call. Getting full value out of this analysis requires AI. You can only look at about 10 markers at once before you start getting very confused. And if you turn on all the markers at the same time, the slide turns white. So it really does need AI or very strong a priori hypothesis. We're also using it to study endometriosis, where the cells of interest are rare and they really do need spatial context. And this is work which I'm doing with Paul Young, and it's led by Aura Lee. And I want to acknowledge the MAPcore team of Anna, Spencer, Rodrigo, and Alan, who've worked with Aura Lee and others to get all of this up and running. So this is MAPcore. We exist for the community. We're available to work on anything, really. We're hoping to develop our own tissue processing so we can better control pre-analytical variables. But right now, we're doing histology, IHC, laser capture, and then we're doing both spatial transcriptomics and cyclic IF. The 3D microscopy is also something we're wanting to bring online. But we can offer a lot of technical and pathology expertise in addition to just doing stuff as needed. And it's led by Dr. Seti Boromand and a group of researchers and some of the leading pathologists in the country. So on the other side of the matrix, we have OVCare. So if MAPcore is a core facility which exists to serve many research teams, transplant research, brain researchers, cancer researchers, OVCare is very specific. It was basically a response to a call to action from Diane Miller, who 25 years ago, who basically declared that she was seeing sicker women dying of ovarian cancer and felt if we worked together, we could do something about it. So Blake, Diane, and I met and thought, well, maybe we can. And we decided to form a team to create core resources, to share them and try to co-opt people to working on ovarian cancer. This was a great success. It far outstripped what I thought we could do. First successes included us showing that ovarian carcinomas really just aren't flavors of the same thing. They're distinct diseases which need distinct solutions, which led the way for subtype-specific research and care. No one would do ovarian cancer trials of all subtypes now. And then we made some key discoveries of mutations in subtypes of ovarian cancer, which became diagnostics. Sadly, haven't moved far beyond that. We also established the genomic connection between endometriosis and cancer, and this is work we're continuing with Paul Young to better understand why the symptomatology of endometriosis is so much more severe in some patients than others, and why some patients respond to hormonal and other treatments and others don't. Developed opportunistic salpingectomy as an ovarian cancer prevention strategy. But then we found that we were working a lot, and Jessica McAlpine developed really what has become the globally leading endometrial cancer team. And we had Amy Jameson and Lynn Hong with Jessica Blake and others doing similar work in vulva cancer. And so we weren't really ove care. We'd sort of lost our sort of ove specificity. And so we did a very BC thing. We thought, well, why don't we just break down the walls and start working more with people? Because then we could connect better with the fantastic researchers at BC Women's, such as Gina, Laurie, and Nicole, and others, and form the Gynae Cancer Initiative. As I get older, I need greater levels of stimulation to get me really excited about stuff. And I find that the idea that if we work together, we can decrease death and suffering from these cancers by 50%, absolutely motivating and exciting, and something I'm willing to drive hard until I retire. Um, we're led by the team on the left, but most importantly by Michelle Wu, who's supported by Vivian Lack and Julian McLeod. Our mission requires efforts across all gynecologic cancers in prevention, detection, treatment, and survivorship. So there's a lot of work to do, and it has to be done by many people. So we really need to impact our whole population if we're going to make that difference. We can't have this as an ivory tower exercise. And we have to work across gynae cancers, across the spectrum of types of research. And so there's so much room for people to build their careers within the team. There are so many questions, so many important things which need doing. And we also have to engage with many stakeholders, including patient partners. And Gavin Stewart was particularly helpful in us kind of pushing out beyond the academy and connecting with people across the province. So, well, my own research now, I'm going to shift into a more selfish focus. So, where do I exist within this matrix of activities? So, when I started studying pathology, histotypes of ovarian cancers and gynae cancers were just really thought to be cool patterns. They didn't change any management. And we managed to show that each histotype is a distinct disease, which is progress. And then we did a bunch of work, as I mentioned. We found the key mutations in a bunch of these ovarian cancer and other gynae cancers. And we got very excited about it, but learned the hard way that mutations really aren't the answer. So we found that each histotype is defined by distinct mutations, but we find mutations in endometriosis and also normal endometrium as well in a lovely paper by Vivian Lack from a few years ago. Um, so mutations don't make cancers, and there are some mutations which are really hard to sort out. So one of the things which came from next-gen sequencing is the many, many mutations which impact the epigenome. And of course, cell context is defined and maintained by the epigenome. And so the impact of mutations and drugs that perturb the epigenome really have to be studied in very strict cell context specific models, which just didn't exist. And so those mutations have been very hard to work with in terms of turning the discovery of very common mutations into treatment opportunities. So we had to move beyond there in our thinking. And around the time COVID hit, I had started to see cancers not as the products of mutations which occur in cells, but reflecting that each cancer type is the endpoint of a distinct oncogenic journey. And like in any journey, where you start with the cell of origin, the terrain you cross, the microenvironment, and what happens along the way are really important. And it's the interaction between these factors which make the specific types of cancers which are manifest by what we see clinically. And if you change any of these variables, essentially you end up with a different cancer. And I put a little house in the middle. This is my intellectual home. This is where I think, this is how I try to think about everything. And it's the perspective which I bring to the table when I work with collaborators. So let's start with cell of origin. Where do high-grade serous cancers come from? What was curious is high-grade serous cancers were not really related to any other serous tumors of the ovary. Benign serous tumors were really stromal tumors. Borderline tumors were connected to low-grade serous cancers. Then you had high-grade serous cancers. So if the first two types were ovarian tumors, the other one rarely presented in the ovary alone. The explanation for this, or potential explanation, was raised by Louis Debeau in an editorial, which suggested that these cancers did not come from ovarian surface epithelium. Now, Louis's suggestion that they came from the secondary Müllerian system is probably was wrong, but he did make people start thinking. And then Jürgen Peek, with colleagues from Holland, were looking at fallopian tubes and ovaries from women with BRCA mutations and found the first credible precursors for this common cancer in the fallopian tube and not the ovary. These are these stick lesions. And since then, this has been recognized as the source of most, if not all, high-grade serous cancers of the ovary. And how big the most is still an area of debate. Since these are described, people have started to look at fallopian tubes. And in the high risk scenario in BRCA1 and 2, people process the whole of the fallopian tube carefully. But in low risk scenarios, fallopian tubes were either not looked at or just shoved into one or two tissue blocks. And we know that in the high risk scenario, when they've cut through the tissue blocks from these CFIM protocols, they double the number of sticks and other lesions they see. And so we know that in the low risk scenario, we're missing the vast majority of these precursors, but you can't really make a health economic case for doing exhaustive analysis because the risk is really so low. A lot of work's been done on this. People have found clonal relationships between these precursors and cancers, more sophisticated clonal hierarchy studies have done, which show that the fallopian tube lesions are usually right at the beginning of the cancer journey. And this work's ongoing along with the development of model systems. What was unique about the BC response was very much flavored by Diane Miller, who's very pragmatic, was saying, let's think about how we can help use this to help the general population. Blake and I were getting very into the idea of something which turned into the tuber wisp trial, how we got to do something like that. But she said, what about the general population? And we realized that quite a large portion, about 20% of women in our population who had these cancers had had procedures where fallopian tubes could have been removed. And so Diane said, well, why don't we just start removing more fallopian tubes? Let's remove them at the time of hysterectomy and remove them in place of tubal ligation. And she coined this opportunistic salpingectomy, which really is a huge thing from BC. There were obviously major questions. Is it possible to change practice? We didn't get grants funded initially because people didn't think we could change practice. Is it safe? And this work has been led by Gillian Hanley with Janice Kwan and others from the group. Will it work and to what degree? And this is the key thing. So if we're going to change medical and legal equipoise, you've got to show that it's useful. And we would hypothesize that fallopian tubes removal will prevent cancers spreading from or through the tube to the ovary. Fallopian tube removal shouldn't prevent the transformation of pre-existing endosalpingiosis, endometriosis, or ovarian surface epithelium, if that's a source. Fallopian tube removal should not prevent ovarian-derived cancers, and OSC-derived high-grade serous cancer should dominate over fallopian tube-derived cancers in women who've had opportunistic salpingectomy. And we do not expect opportunistic salpingectomy to prevent all high-grade serous cancers for a whole bunch of reasons. But one of them is that these opportunistic salpingectomies are done in low-risk scenarios and is not effective from a health economic perspective if the surgeon spent a lot of time trying to get all of the fallopian tube and large bits are left behind. That even happens in the high-risk world. So this is a very strange image. What it is, it's a piece of ovary removed in a woman who had a salpingectomy, then afterwards had an oophorectomy as part of sort of tuber wisp. And you can see a huge chunk of fallopian tube was just left behind and stuck onto the ovary. So if that happens in the high risk scenario where we know the fallopian tubes really have to be removed, we know it's going to be happening in the community. But things don't have to be perfect to be really, really useful. these are the later data, which is coming out in JAMA Open Network. And of course, this work is led by Gillian with my team and a host of other collaborators from around the world. And it basically shows that in removing the fallopian tubes at time of other surgeries reduces the risk for these cancers, which are the serous cancers, by 80%. And we see no effect in breast cancer in this study. As that's an incredible shift. And we don't know over time whether that actually that number could get the 80% could get higher as the cohorts we're studying age. So what's going on? The if we along with looking at the population data, which is Gillian's expertise, we went to pathology labs around the world and we said, if you ever have high-grade serous ovarian carcinomas come in to your lab where fallopian tubes have been previously removed, please tell us what types they are. And so on the left, you got the standard distribution of ovarian carcinoma types. If, as we from our population data, you removed 80% of the high-grade serous cancers, you'd be left with that piece of the pie which is still covered in the second image. And if you expand the pie, this is the new distribution if you've removed 80% of high-grade serous cancers. High-grade serous cancers would still be the most common type. What we see from around the world from ovarian cancers post-salpingectomy is the pie at the other side, which really does match this 80% reduction. So I think this is real. I think that the new paper coming out should shift medical equipoise and at some point legal equipoise, which may be more important in the States. But I should acknowledge that whereas in BC, because of the fantastic connection between the gynecologic oncology community and the general gynecology community through the DA Boys Society, they were able to really, Diane could just say, please, will you consider doing this? And it started happening. There are different barriers to this surgical practice change in different jurisdictions, including some major barriers in terms of coding, which exist in the States. So can OS reduce the incidence of ovarian cancer? Yes, it can, but the numbers of surgeries which are being done are decreasing with time. So the opportunity is slipping. To address this, Gillian has been working with Heather Stewart and many others to see if we can expand the opportunity to other GI surgeries, and they're leading a clinical trial on this. So this all started with observation of a potential precursor lesion 25 years ago. It's really quite amazing. It's like a low-tech discovery using 1850s technology has led to this massive shift. In BC, high-grade serous cancers of the ovary have been decreasing in incidence for a while. And this is not due to opportunistic salpingectomy. This all happened too early for that. What it's probably due to is the profound impact that oral contraceptives have on reducing ovarian cancer risk, where 5 years use of oral contraceptives provides a lifetime risk reduction of about 50%. That is huge when you consider the number of women who take oral contraceptive pills. We started looking at this superficially, and we found a collaboration with Gillian and found that in individuals who take oral contraceptive pills, when we look at their fallopian tubes, and in particular fallopian tube precursors, we don't see less p53 signature lesions, which we thought we might see. But when we see them, they're much smaller in the women who have taken oral contraceptive pills. We wanted to have a more systematic and serious look at this issue because, as this community would know, OCPs are being used less and less, and more individuals, particularly younger people, are opting to get LNG IUDs. And yet, ovarian cancer is still lethal for most patients. OCPs provide protection. We don't believe the IUDs do provide any protection, or if they do, it's minimal. And there are new options such as the dospirenone or only contraceptive pills, and maybe they could prevent ovarian cancers, but it's going to take us 30 years to get those such data. And so we decided to ask the question of how did birth control pills prevent the cancers? Do we see the same effect from IUGs or progestin-only pills? And then start thinking about how we can use this information to prevent there being a sort of epidemic of ovarian cancers in 25 years' time, because it's still an untreatable disease. And we formed a really interesting team. We were supposed to have heard the results of our team grant quite a while ago. This week we heard it's been delayed again for another week, even though the grant was submitted in June. We called it the Beacon Study, and it connects uh, sort of lab folk like myself through populations to the incredible work of the CART team led by Wendy Norman and others. And suddenly our patient partners are through Planned Parenthood, which is really exciting. And the way which any of our findings could translate will also be quite unique as well. I'm going to show you some data we've got from our preliminary data for the grant, which is really quite exciting. And this is a grant which, whatever happens with the current funding, are we going to get this funded one way or another? It's too important. And I note that you wouldn't be able to do this work in the States. So how to look at, we have 7 different groups of exposures, pre and postmenopausal individuals. And so 100 patients from each, that would be 1,400 fallopian tubes. So how can we study those in detail? And this is where doing the sort of spatial analysis can really help, because we do it on tissue arrays, and suddenly we can amortize the cost over about 40 patients in each analysis. This is just our preliminary data tissue array. It only has 20 cases of OCP users versus non-OCP users who are postmenopausal. And we didn't really expect to see much other than being able to show, hey, this works. But in the preliminary data, we can see we got a very large number of fallopian tube epithelial cells. If we look in the epithelial cell population to do a volcano map between OCP and non-OCP users, we see there are a bunch of interesting genes sort of arise as being significant, including the family of serpin genes, and as being expressed more in OCP users. And it's not so much that they're expressed more in a cell type, is that it turns out there are different populations of cells. And if we look at the bar chart on the bottom of the right, we can see at the bottom there are different frequencies of subpopulations of fallopian tube secretory cells. There's the non-OCP users have more what we're calling type 1, and the OCP users have more type 2 and 3, and the type 3 being marked by the serpin family of genes. If we look in published data sets for which compare pre to postmenopausal fallopian tubes without reference to contraceptive pills, we see the genes which we're seeing in the OCP users are more found in the secretory cells in the premenopausal group. So this has changed our hypothesis to one which I think sounds to me credible and really interesting, which is, do OCPs prevent the cancer by delaying the maturation of populations of fallopian tube secretory cells from a premenopausal state towards a more cancer-prone postmenopausal state. And once we know what's going on, perhaps we can find other ways of preventing it. So that's a little bit on cell of origin. Shifting to mutation, which is obviously important. And we found mutations when we did our next-gen sequencing. One of the mutations we found was in our studies of Sertoli Leydig cell tumors of the ovary, which are really interesting because essentially you have testicular tissue growing as a tumor in ovaries. And this work is led by Yemin Wang in my team, who's now established as a new investigator. So these cancers occur at a variety of ages. They can occur in much younger people. They can be high grade and low grade. And what we found is these mutate cancers often have mutations in the microRNA processing gene called DICER. The mutations are always in the same places. They impact metal binding sites in the RNA's 3B domain. DICER has been shown to be important in these cancers because if you have germline mutations in DICER and have DICER1 syndrome, you're at risk for pleuropulmonary blastomas, but also Sertoli Leydig cell tumors, and a range of other cancers, including 3 types of kidney cancer. So our mutations are interesting. There's not loss of heterozygosity in the usual way. You have one copy of the gene is lost, which is germline, and the second copy you have these very specific missense mutations. We're able to show that what these do is lead to a complete shift in microRNA processing, where microRNAs have 2 sides, a 3p and a 5p strand. This stops the processing of the 5p strand microRNAs, and you only get half of the microRNAs processed. We were able to take this and improve diagnostics, but we were not able to do anything in terms of improved treatments because there weren't any model systems. So Yemen, we developed this, the first transgenic mouse for this syndrome. Unfortunately, the mice went from healthy to kind of blowing up and being too sick to study so quickly we couldn't really work with it. And so we started working with Mike Underhill, a researcher from UBC who studies mesenchymal progenitor cells, and he had a mouse model which targets those, and so we refactored the model so we could put these combination of DICER mutations into this model. It's very a complex mouse, but basically when we give the mice tamoxifen and all the HIC1 mesenchymal progenitor cells, they knock out one DICER gene, they put this missense mutation into the other, and in those cells they also express TD tomato, which is a lineage tracer. These mice have a highly penetrant but rather delayed phenotype um, of renal tumors. And when we look at the renal tumors, looking at TD tomato, we find that the stromal cells are the tumor. The epithelial cells, which are often seen in DICER1 tumors, they're drag-alongs. We find the same mutations in these cancers we see in human DICER1 cancers, such as KRAS and P53 mutations. And then we started asking, well, where do these cancers actually come from? And we have a paper led by Yemen and the team, which shows the answer to this. So in the normal cells, we see occasional they're in yellow, these fibroblast progenitor cells, or very, very early universal fibroblasts. In the earliest DICER1 tumors, they predominate, but when the tumors turn to a high-grade anaplastic mess, they're kind of lost. We're able to do mapping of these cells in normal kidneys from these mice, and we find that this cell population occurs in normal kidneys exactly the places we see the earliest tumors, which are cortical, just under the surface of the kidney, perivascular, and also subepithelial, in particular at the hilum of the kidney. So we now have a model system. We're starting to do tumor suppressor genes and looking at candidate genes for mutations, and this is in press in Nature Communications. But things aren't moving fast enough. So this is where it comes back to MAPcore, and can we use ADCs? And so we're starting to screen our DICER1 cancers, and we've got a bunch of cancers from the DICER1 consortium. Jonathan Bush, who's on the call, is key in this. To start saying, do they express any ADC targets? So could we move quickly into some clinical trials for these disease cancers because they really need them? Now, considering this model, we have a mutation, but there's weird stuff going on. We see kidney tumors. Why don't we see tumors in any other organ, like Sertoli Leydig cell tumors or pleuropulmonary blastomas in this model system? Because the HIC1 cells are expressed everywhere. So there really is a microenvironment or anatomic factor here which we need to understand. Now, talking about microenvironment itself, endometriotic cysts in the ovary are perhaps one of the harshest microenvironments which exist in humans. And 2 cancers come from this, clear cell and endometrioid ovarian cancers. And these cancers are, this has been known for a long, long time. These cancer and the epi data supporting endometriosis and ovarian risk has also been known for a long time. In particular, this is a fantastic paper which really proves it. So how do these 2 such very different cancers come from the same precursor? Initially, we thought they would have different mutations, and it turned out they don't. They have different frequencies of mutations, but there's nothing which is mutually exclusive. We then started thinking, well, could there be something different in the something different about cell state or response to microenvironment? And we did some proteomics on these cancers several years ago and found that clear cell cancers express something called cystathionine gamma lyase. And this is a protein which is key in the transsulfuration pathway, which produces glutathione when glutathione is needed to detoxify cells, which are in really nasty microenvironments like an endometriotic cyst. We found that indeed this is expressed in clear cell cancers of ovary and uterus, but most interestingly, it's actually expressed in ciliated cells of endometriosis and in the uterus. And this is something we want to work with Maddie Salker on if she's on the call. In clear cell cancers which come from endometriosis, we see it's expressed in the endometriosis, expressed in the precursors, and expressed in the cancer itself. CTH seems to be turned on when you expose endometrial cells to harsh environments like an endometriotic cyst, including exposure to endometriotic cyst content. It also looks like it may be a good target in these cancers. If we knock CTH out in cancers, they don't metastasize as much, they don't grow as much, and they seem to be quite vulnerable to other treatments, including mTOR inhibitors. So this does seem to be the cells, the microenvironment uh, encourages the growth of cells in a particular cell state, and that cell state includes the expression of CTH. These cells might be like early secretory phase endometrial cells, which also express CTH. And then if they transform into cancer, they just carry this feature forward. So it's a microenvironment induced feature, which leads to the expression of CTH. Then we started asking, what does CTH actually do in these cancers? It turns out CTH seems to be key for the expression of HIF1 alpha, which is one of the key ways which cells can regain vascularization and deal with hypoxia and such microenvironments, in addition to the creation of glutathione. And in looking to see how CTH, which is really just known as an enzyme, could contribute to the production of HIF1 alpha, we were really surprised to find out that it seems to be connecting directly with the translational machinery, where CTH actually seems to bind to the HIF1 alpha message, and more importantly, to some of the translational machinery, including EIF4F. And we're trying to finish up this story, but what we end up with is something where the microenvironment itself encourages the growth of certain types of cells, which, if they transform, carry those features forward, and they add to the aggressive nature of the cancer. And this occurs through two ways. One is through the known kind of canonical production of GSH, but also through this other factor, which is that it may directly connect with the expression of HIF1 alpha and other proteins. So we're exploring CTH now as a therapeutic target in these cancers with Art Chernikov's group, and Lucy Lee is leading this work in the lab right now. So I'm going to end the talk here. I'm sure others could give a more coherent demonstration of how programs and cores interact. But I'd like to thank the patients, funders, trainees, team members who make our dreams of having a positive impact possible, and you all for listening to this lecture early in the morning. I'd be very happy to address any questions you may have, and of course meet with people afterwards if there's anything which interests you, or if there's anything I've discussed which you think could be useful for your work, we'd be happy to share.
Thank you so much, Dr. Huntsman. That was an amazing journey through an incredible body of work. Thank you so much. I'd just like to open the floor to questions. We have a few minutes here. You can unmute yourself or pop something in the chat, and I'm happy to read it out.
I'm going to jump in first. Thank you so much, David. It was really wonderful to see the full story all in one presentation, and we really should have given you a half a day to go through that. One of the things I wanted to ask you about was just how critical the relationship between yourself as a basic scientist and your collaborators, the clinicians, the patient partners, the other investigators who are doing clinical research. You sort of wove that throughout your presentation, but I'm just wondering if you can say a little bit more. And there's a little bit of a selfish reason. Part of our new strategic plan at the WHRI, in fact, our number one priority is around really fostering these connections between basic scientists and the broader women's health research community. So would love to just hear you talk a little bit
So
about that.
I think this is really important actually because I mean I think we can all take pride occasionally in things we've done and we can say, oh, I contributed to this. And then there's the dumb luck stuff, and you have to be careful not to get full of pride about it because you just got lucky. So we were very fortunate that Diane, Blake, and I uh, could trust each other, complement each other, and work well together. Um, the Diane had a very particular role in which she was a facilitator. She didn't actually engage in research herself, but she didn't get in the way of it and try to, you know, direct. And that worked. If we had three different people, it probably wouldn't have worked, or one of us was different. So these teams really have to be, there has to be compatibility and trust from day one. We started with a problem. We didn't aggregate around a source of funding. And I think if the money comes first, you end up with people who just want money, and there'll be a team of convenience, and it won't last. And so if you encourage teams to coalesce around problems and then support them, you're probably more likely to get people who aren't just showing up in the room because they think there's a pot of gold. Hoping that kind of, I'm happy to have a coffee with you and discuss more. Hi, Wendy. Oh,
Thank you so much, David. This was just absolutely a whirlwind. By about 10 minutes in, I realized I'm going to have to sit and watch this again, and perhaps slow down and just be able to take in all of what you've covered in this amazing journey that you shared with us. And I think one of the things that really stands out for me is this sense of being able to harness the curiosity and to do it collaboratively with others so that you appear to have presented what looks to me like a master class in engaging others in thinking about what could be our next steps of the problem. And, so many of your different slides brought up, and we saw this, and then that just didn't make sense. And so and so had an idea that we should go here, and we followed that. And I can only think that around those tables, lots of ideas are coming up, and that partly from what I've seen since we've been collaborating together, is you seem to have an just a bit of a genius about which of those strands to follow, which ones are going to probably bring together your clinical expertise, your lab expertise, but there's also something else in there, I think, that's really been able to define which are these threads that are going to make a big difference for us. Do you have an internal key that helps you to decide where to go on those? You've got lots of different choices to follow in front of you. How are you zeroing in on those ones that are just being so impactful?
so there are some really wonderful aspects of the academy in BC and how it connects with the healthcare system, and that creates very specific opportunities. There are other parts of us which are very mediocre. And so when we look at the plethora of opportunities in front of us, I try to focus on the things where we have a unique competitive advantage. That sounds pretty harsh, but doing smaller versions of studies, which could be better done in Boston or New York, really doesn't hold any interest for me. And there are some unique opportunities. Like the Beacon study we're talking about is brilliant. You can't even do it in America right now. I mean, they would laugh at a grant doing that, but it's so important. And we're a small community. We can, as So sort of some geeky molecular pathologist person and Yongjin Park, a causal inference researcher, we can work with CART, with you, and start addressing this. So that kind of unique kind of coalescence of energy which can occur here, the escape, one of the, there are not just a healthcare system, there's something I think unique in the BC culture, and I don't really understand it, but the escape velocity is much lower here than in other places. So sustaining things is difficult, sometimes impossible. So you have to always expect your sandcastle will get knocked down and just have to build it again. But the people can just coalesce and do stuff. And it might be because others are too lazy to stop them or something, but there's nothing stops teams from forming and doing interesting work. And I think that is a key thing. So if you can find some unique way of looking at a problem where you bring other people to the table who just can't, where it can't happen in other places, that itself creates a competitive advantage.
Super. Well, thank you again for just a wonderful presentation, but also a brilliant career, very inspirational. I'm so glad to see so many of our early career researchers and up and coming trainees here.
I think we can probably squeeze in one last question if anybody has any final thoughts.
I'm conscious of time, but I just really wanted to thank David and echo Wendy's comments. I think many pearls of learning, both from the actual content, but from your ability to create team science and some of those thoughts around don't always think of the wicked problem and then think about, are we the right team, the right place to try to answer that? And that doesn't always marry, but when it does, it's magic. So really, really want to thank you for your contributions. And I'd love to offline with you about the microenvironment and the role of the microbiome envirome in that microenvironment at another time.
We almost put it in the Beacon grant, but we ran out of money, so it'll have to go in the next one.
Yeah, well, let's brainstorm around that.
Okay. Well, thanks so much for inviting me
Thank
to join you
you.
this morning.
Thanks, Dr. Huntsman. Thanks, everybody, for joining us. Have a great day, everybody.